the reason for this title is actually that this problem has been puzzling not only me, but me too, for now over 10 years. And over 10 years, we have treated this problem as a puzzle, looked at here, looked at there, in the hope that one day we will come up with a bigger picture. And I don't think we are really at that point, but I do hope that I will show you what we have done over the last 10 years and how we contributed to this field. So the work which so in a sense, what where I come from is probably from a school of something which you can say the bottom-up approach. And the idea of the bottom-up approach is to take some biological problem which is not fully understood, hopefully, and then try to isolate the main ingredients, or at least what you believe are the main ingredients, and then shovel these main ingredients into a kind of a mimetic system. And then you kind of try to use your physics and this knowledge and physics tools to understand this mimetic system. If you manage to understand this model, then you can kind of increase the complexity of the model and hopefully give a feedback to biology, and in what the funding agency particularly like is to design advanced materials and processes. Now, how much is this really successful? That's, of course, subject to debate, but that's the debate that you all know. And then you play this game in a circle, in a circle, in a circle, in a circle, and you hope one day, you know, either your problem becomes non-trackable and you get lost, or, you know, you make a nice contribution and then you have understood something. All right. So what's the principal task? The principal task is to use analytical and simulation techniques that are typically developed in the statistical and soft condensed metaphysics, and then apply them to this biological problem. Try to come up with effective Hamiltonians, use hydrodynamic equations. We have heard a lot about it this morning in a fairly formal way. I'm going to do the nasty thing. I'm the physicist approach. I am not doing it as nearly as elegant, so I will apologize to mathematicians for that case. In principle, you're treating the system from atomic levels to cellular components and tissue devices, and in all of that, the only way to go through this forest is to rely heavily on your experiments. If you do not have an experimental background, at least in my own personal opinion, it is very hard to find the appropriate parameter spaces. It is very hard to say, okay, I can drop these terms in these types of equations, and that can help you a lot. So using the experiments is, I think, a vital part in this process of modeling. All right, so what I will do today, I will present the work of Timo, Mislav, and Daniel. Mislav sits there at the back, and he is doing the fusion. So for the parts of the talk which are related to him, you're most welcome to talk to him too. And this whole project has started, as I said, or was started by my scientific fathers, and they were Udo Seifert and Erich Sackmann. Udo taught me the arts and crafts of theoretical physics, and I have learned enormously from the experiments of Erich Sackmann and his group. And there were another two people in this group. So I was a PhD student, Kea was a postdoc, and Rudi was a habilitant. But we never collaborated while we were there. We started to collaborate many years later. And this has actually created a very successful collaborations with two students which have graduated in that and which produced enormous amount of data, this wealth of data which we as theoreticians could use to model, and I will show you some of this data today. Okay, so what are membranes? We have heard a lot about membranes from the mathematical point of view. We treat them as two-dimensional objects which are fluid. They tend to be not stretchable. They have very beautiful properties as materials. I will skip that. I will talk about them a little bit in the biological context. Membrane in a cell is an organ. It's a sensory organ, right? At least the plasma membrane which surrounds the cell over here. the cell uses this organ to sense its environment. And that is something which the cell basically needs. So it is one of the key steps for the cell to have a membrane. Most cells, or let's say 99% of the cells actually have a phospholipid membrane because it helps to compartmentalize. So the cell knows its end. But the membrane is not only here at the end. The membranes are all over the cell. All of this. There is a nuclear membrane, then the endoplasmatic reticulum, which is a phospholipid membrane decorated with proteins, Golgi apparatuses. There are many, basically, mitochondria. Basically, this concept of compartmentalization is not only on a cellular level, it's also on a subcellular level. All right. Now, it's what kind of environments this cell live and work. They work in an extremely noisy environment. There are millions of processes happening in a cell at the same time. The cells have to, these membranes have to respond to that. They tend to be out of equilibrium, otherwise it would just somehow be dead, and that would be somewhat boring. Membranes are also crowded in the context that there are lots of proteins on the membranes. So these proteins have to find a way in order to make an action. This protein has to find a way from this end to this end of this little vesicle, and it has to find its way through this entire forest of other proteins, and of course this is a crowding problem. There are also competing processes at the same time. The cell is investing energy in one process, that means that it cannot invest in another process. And how this happens then, this is really a question. So obviously, if you look into something like this, you have a promise for very rich physics on multiple timescales and length scales. And if you think about it, really, I mean, cell does its job for, I mean, since the cell exists, it does it in a very reproducible, in a very controlled manner. It relies on these proteins going somewhere, interacting with another protein, then it's producing a signal. And it does it very reliably. And the question is how and why. Why these proteins don't get lost? How these proteins know where they need to go? How do they do this? These are all actually very fascinating questions, at least to me. So, if I think about membranes, of course, in the cellular context, then it is very hard, because it's not only the membrane coupled to other processes, plasma membrane couples to the cytoskeleton or some sort of cytoskeleton, depending on the type of the cell. Inside of the cell, there are a number of other structures, a number of other things which interact with the membrane as well. But if I look at all of these things, in the membrane, outside of the membrane, the interface of the membrane, and the inside, there are some things which I will always find if I think about membrane-related processes. I will find the membrane. That's number one, right? That's the point. This membrane will deform and fluctuate somehow. The deformation of the membrane can be either driven by the healthy Hamiltonian that we have seen this morning, or by a more complex Hamiltonian for the bending deformations. But in principle, I have a membrane. This membrane deforms and fluctuates. This fluctuation can be active, can be passive, can be whatever, but the process is here. Another thing which is always there is proteins. These proteins float through this membrane in some kind of a way, and they will interact with other proteins. So I have molecular complexation in confinement of a membrane. It is not the same to have one molecule here and one molecule here in solution, and they float all over the place. They meet and bind and unbind and so on. If you do it in confinement, of course, I can't go and meet anybody there to shake hands, right? I need my confinement to move with me, and then I can potentially go and shake hands, right? So, your complexation is intimately related to the confinement, and you cannot describe it without the confinement. And of course, this, as I said, proteins have to move from one side to the other side, and in principle that leads to driven passive or obstructed transport, or there are molecular motors which drag the whole things across in some kind of a way. So now, I will look at this, and if you think about it in a sense of physics, what are here written are three stochastic processes, which works on different timescales and length scales. So what I look is, in this talk, I will only confine myself to bending elasticity. I will look at molecular complexation in some kind of an average mean field way. So I will not look at into atomic resolutions. I will not know which protein it is. I will describe it in a fairly coarse-grained fashion. And I will only look into simple diffusion. I don't know, but this doesn't really work anymore. All right. No?
If you can use.
Huh? Yes? Yeah.
Oh.
All right. So now if somebody asks a bad question. All right. So if I think about an experimental system which brings these three processes together, then it is a decorated phospholipid vesicle binding to a decorated substrate. All right. Now, from this point on, I need to know who is in this public. So how many biologists are here? Okay. How many physicists? And how many mathematicians? Okay, 1/3, 1/3, 1/3. I'm gone, lost. I can't do it all. So let's do it like this. Please ask questions, right? I can't. It's very hard to kind of cover the three fields in some kind of a way. I'm not even as good as all of this. I mean, I'm a physicist, so obviously I have limitations in biology.
An immediate question is, I mean, this apply, does it matter in 2D and 3D?
This is in principle in 3D, and yes, it would matter whether you are in 2D and 3D. Now, of course, elasticity in 2D and 3D are completely different things. That's one thing. Now think about transport. It's very easy to block in 2D. In 3D, you can go around. Right? In 2D, this collapses into a 1D problem, and transport collapses in 1D problem, and in 3D, you still have a surface. So even if I have something, I have this. So yes, it is important whether you're 2D or quasi 2D or quasi 3D. All right. Now, how does this look in an experiment? In a typical experiment, the master of this experiment is here. So, you know, if I say something wrong. So this is how this vesicle looks if you look at it from below into this region. So I'm not looking this way, I'm looking this way, up. Okay? So this is where the vesicle contacts with the surface. And this is pretty much real time, but you see on a microscope, and you see that this and what you see here is a reflection interference contrast image, which means that these patterns change as the membrane fluctuates above the substrate. So for the physicists, this is equivalent of Newton, dynamic Newton rings, right? For the
so the membrane is really sitting on a substrate.
This membrane, the vesicle, has come down to the substrate. It has spread to the substrate in some kind of a soft potential. It hovers in this soft potential at some finite distance from the wall and fluctuates. That's what it does. So when it does these fluctuations means that they are changing height. And the beauty of this technique, so what you see is basically a light which reflects from the membrane substrate interface and the membrane glass interface, and you get this interference pattern out below, and this is what you see dynamically. All right? And now you can analyze this image, and you can convert this image into something which we called a height map, right? Which tells you, okay, as I move along this surface, I can see where the membrane is high and where the membrane is low. Or you can you can calculate a dynamic fluctuation map. So this tells you that at this point over here, the membrane height is at the particular point, but it fluctuates with this particular amplitude, right? Also, what you see is there's much more structure in the fluctuations than in the height maps. That is because there are some tricks which you can use over here, and that actually increases the resolution of the system. If you want to know how to do it, please ask me or Rudy, or look into this paper, which we kind of explained that. So, now this was only the membrane. There were no proteins in this membrane. All right? Now what happens if you put the proteins? I'm making the movie 10 times faster, otherwise we'll be sitting here and only watch my movie. So, this is what happens. You see how these plaques nucleate, and then they grow, and they form a domain. All right?
What kind of protein?
So here we are talking into E-selectin bind by E-selectin deabsorbed onto the surface binding to S-lex cellulose X, which was put into the vesicle membrane. All right? And what you see is that you have this E-selectin which are waiting for these ligands which are in the vesicle to come from the bulk of the vesicle, bind and create a domain looking like this. And you see that this is a fairly dark domain, which also tells us that they're quite tightly packed. Okay? All right. Now, this is in principle the protagonist of our experiment. And now you can change the proteins, you can change many parameters in the systems, and then what you get is a large variety of adhesion zones. So, large variety of domains. So only by playing with these free stochastic processes you get very different morphology of ligand receptor binding. And what I'm now going to focus on is how this domain emerge, how can I model them, and what makes the system doing sometimes one domain, sometimes 100 domains, sometimes I even have a coexistence of very tightly packed domain and lightly packed domains, or even sometimes I end up in this kind of a jammed ring-like structures which are kind of metastable, but on the lifetime on the experiment definitely stable. So this is what I'm going to try to explain. Can we actually model this? All right, so I'm going wrong way. All right, so if I want to model, I first have to come up with some theoretical descriptions of my free stochastic processes, right? And the first way to do is I first have to describe the membrane. And what you see here is basically what you heard this morning in another representation. This is in a representation when the membrane is basically flat. And it tells you that, okay, this is a famous Helfrich-Hamiltonian decorated by one more term. So what is this? This is basically the bending, and we have heard about the bending a lot. This term accounts for the tension in the vesicle or in the membrane. And then there is this thing which is a non-specific potential. So why do we put this term? We have seen that when the membrane, when the vesicle came to the substrate, it didn't really go all the way down. It somehow stood up and hovered there, right? This is a kind of a continuous potential between the membrane and the other membrane, a membrane and a substrate or something, whatever you like. All right, now, this is what is the additional term that we need in order to describe membrane adhesion. Okay, now, this Hamiltonian has been on the market 20 years, 25 years, all right? However, it was actually never proven experimentally that you can actually use it. And I think the first proper experimental proof where you can actually extract these parameters in some kind of a systematic way came only last year in this work, and this was a lot of sweating and a lot of discussion and a lot of hard work to see that if you treat your data properly, you can actually come with a consistent set of parameters describing your Hamiltonian. So I won't go into details, but just that you know, this is a good model. At least there is a range of parameters where you can use it. That's the point. Now, about this non-specific potential. In cells, people tend to completely ignore the existence of non-specific potentials because they go and they say, well, it's protein-driven. Well, cells don't have membranes just sticking to each other. We don't have a glue of membrane in the cell. And that's because we actually have some things which we call repellent molecules. And basically any protein which is not binding to another protein on a substrate or on another membrane is a repellent molecule. And what it does, it helps keeping these membranes separated. All right? And I think it was Brunsma first who suggested that this kind of action can be integrated out into kind of a non-specific potential. I think this was done in the late 90s. And then basically that this becomes our gamma, our little non-specific potential over here. All right? So such an action enters into this term over there. Okay? And you can see that it really kind of works, because if you increase the concentration of these guys, these repellents over here, which somehow occupy the membrane, and it's actually a steric potential. And if you increase their content in the membrane, then, and you keep everything else the same, then the number of bonds which you form in the contact zone basically drops significantly. Okay, so that is then an action of a non-specific potential. Okay, so so nobody asked any questions, so I suppose everybody is asleep. So is that clear, or does this make sense? Yes?
Could you replace the final term in that functional by just a sum of imagine that the interaction with the substrate is at points, so you could replace that by a sum of delta functions.
Okay, so
located at
yes,
where you might think the
yes, that would be fine if the membrane would not be fluid. So that is precisely what was in a sense done in this paper over here. And you can't really do it by point interactions because these molecules tend to move. So they also have entropy. So you somehow have to account for their entropy and their enthalpy. So the delta function would give you an enthalpy, and you still have to work with the entropy bit.
But I guess that the gamma is a constant.
Gamma is in principle a constant, which kind of says, okay, if I fix this particular height and fluctuation amplitude, that I have entered the steady state, and then I can map one into another. That mapping is highly non-trivial. It was in principle done by Grundmann for an open system, not for the closed system. And there was an attempt in a closed system over here.
But should there not be a distribution? Shouldn't there be some kind of distribution?
Yes, it should be a distribution, but also when you have a repeller interacting with the substrate, this is not fully localized. It also spans a lateral correlation length of the membrane, and then at sufficient density you can kind of average it out.
It may happen that delta function like potential will not let the particles free.
If it's not free, then you couple it, and that would pay entropy. In that case, you really have to take it out.
Finite time of each bond, right?
Yeah.
And you will have infinite time.
Yeah.
But if there were delta functions, you could have. There's the delta functions on the substrate, and there's the delta functions on the surface of the membrane.
It's on the surface of the membrane.
You could think it tethers, I guess, which.
Yes, but you would actually rather like to think about these things as interacting through a delta potential. Okay? That's much more like it. And this is not something which kind of has an interactions and hangs around. It just somehow floats all the way and pays for being there. It pays for not being able to explore its entire structural space. Right?
It's a mean field.
It's a kind of a...
See it as a mean field approximation.
I was just going to ask, what's a typical spacing between those particles?
What's the typical spacing between these particles over here? That will depend on the size of the particle. So if you have a big protein, then you would think about something like, what, I don't know, 5 to 10 to 20 to 30, 40 nanometers.
If they're closely spaced, doesn't the substrate basically react to the sort of integrated field?
Yeah, sure it does.
Right.
And that's exactly what it creates, this non-specific potential. That's the whole point. That's precisely what I'm trying to say, yes. All right, so there we are. Membranes do not tend to stick to each other, so that the potential of this type is always present, and unfortunately you can't just carpet it. It can be modeled in the fairly primitive way by these continuous approaches, and this Helfrich Hamiltonian is a useful concept, so we can move on. Onto the bond. We have to model the bond, right? And now I'll do something which, of course, I'll apologize now to biologists, but for me a protein is a spring, all right? And I make it a 1 degree of freedom. Why? Because the physics of the problem is already in 1 degree of freedom. You can do it with many degrees of freedom, And just your math is going to be becoming more complicated, but in reality, you will not gain a new physical insert. All right? So we have 1 degree of freedom which fluctuates. So this is a thermalized spring in my case. All right? So my spring can thermalize, interact with either another spring or just a point-like object which floats and moves in the membranes. These guys can also move in certain types of the models. And when this one is above this one, there is a probability that they bind, and they will interact, and I give it an interaction potential, and I use the absolutely simplest potential which I can. I use a finite square well potential because I can describe it but only 2 parameters. I need this parameter. This parameter tends to replace the binding affinity, which we'll typically measure in solution. And this is some kind of a size of the binding pocket. Okay? Some kind of a size of the well between which these two things interact. Now, there are some advantages of this potential, and then there are some disadvantages. We will see disadvantages later on. All right. Now, and what I'll try to convince you that the ligand in the membrane reacting with the receptor on a substrate can be basically formally mapped onto a problem of 2 thermalized springs interacting with the short range potential. Okay? And that is a problem which some of us give to our students in the second year physics in classical mechanics to calculate. And what they will calculate the free energy to be bound. And the free energy to be bound has 3 terms. One thing is an elastic term, which is going with an effective elastic constant of the system. Right? So this elastic constant and this elastic constant, if you have 2 elastic constants, 2 springs in the series, this is the deformation stored in it, and that's all. Okay? So this is a simple elastic term. This is what I get when I form a bond. This is the depth of the well. And then I get another term, which again goes with an effective elastic constant and the size of the well. And that term over here is how much entropy you have to pay to form this bond. When you form the bond, you have suppressed fluctuations. And this is how much. All right? So, having a thing in solution would give you this. Confining them to a substrate or into a membrane gives you 2 additional terms which decrease the depth of the well. All right? So these 2 terms over here intimately depend on the properties of the membrane or of the confinement. So speaking of affinities in membranes is basically nonsensical. That's plain and simply it. All right, now if you rewrite this equation in terms of the membrane notation, then it looks like this, and you see again there are 3 terms which are basically the same. And what you have here is what is the spring constant of the membrane, and the spring constant of the membrane goes with the vertical correlation length, and the vertical correlation length is nothing else how much the membrane fluctuates. Right? So the only thing which I have here, this vertical correlation length, replaces this K2, and then you can rewrite this equation poof, straight out. Okay. Now, you can take this delta F and look at it how it depends on the separation distance between the membrane and the receptor, right? Because we said that if I'm far away, then the probability that I'll bind is very little. If I'm close, the probability is much higher. And you can have a look over here. I put gamma, but you may as well just write the fluctuation amplitudes. All right? So how does this binding affinity depends on the fluctuation amplitude and the initial separation between the membrane and the receptor. And then you can see, okay, well, you know, for very little difference between 25 and so 50 nanometers of difference, and then, you know, basically this is changes in fluctuation amplitude of maybe 10 nanometers will give you up to 10 kBTs. Okay, this is your energetics.
Question.
Yes?
Somehow this fluctuation component
Yes.
seems a little bit strange because it looks like you're correlating the motions. You don't have two independently fluctuating springs. It looks like from the term that they're actually correlated.
Well, once you make a bond, then they fluctuate together.
Oh, but there must be a fluctuation also when they're uncoupled.
When they're uncoupled, they're fluctuating independently. Once you couple them, then you pay 1 degree of freedom,
Okay. So
and you fluctuate.
only component, sure.
And you pay 1 degree of freedom and suppression of fluctuation, right? Yes.
Membrane undulations, I mean entropy of membrane undulation.
Beg your pardon?
Undulation forces are somehow in.
Yeah, sure. Basically, the way you calculate this free energy, you basically calculate the partition functions for the unbound state and the bound state for the ligand and receptor to be in the bound state within the potential, and outside of the potential you divide them.
The membrane undulations are there.
Yes, and they are hidden in this parameter over here. This is the membrane fluctuation amplitude. Right. Okay. Now, and you see that, you know, if you change this height and if you change this, we can go from completely stable bond of minus 5, 6, 7, 10 kBTs to something where you have to pay energy to form a bond. All right? And that is how you can control adhesion, actually. And now you can run simulations with something like this in mind and see where is the transition between bound and unbound state. And that's a line which you measure. Okay? Now, does anybody has a problem with this line? I'm not asking the seniors, I will ask the students. What's the problem with this line? It's way too high. So it basically tells me here, this is stable, this is unstable. But it basically tells me like, aha, I'm stable and I have to pay 6 kBT to form a bond. All right? So that means that my theory sucks. Right? It's not good. I missed something. Okay, so what did we miss? We missed cooperativity. All right? So, if I have a bond on position 1 and another one on a position 2, they will deform the membrane together. They will share the load. They're actually connected by this elastic medium. It's not independent. If I'm very close, the deformation is much different than if I'm very far. And you can calculate this free energy now. I'm looking it onto a lattice where I'm changing the spacing between the bonds. And I can calculate this free energy as a function of the depth of the well. And you can see that you can go from something, have domains now, which are fully stable, that they are stable, unstable, and stable, or for a large part of the phase diagram, simply unstable. Okay? And you can calculate this free energy. The calculation is somewhat more involved, and you wouldn't give it to your students in the classical mechanics class. But in principle, you recover all of the terms again. You have the elastic energy over here, which is now shared over all of the bonds. This is only the elastic term. This is the depth of the well. And that here is a non-local term accounting for suppression of the fluctuations over the entire sheet. All right? So, the prediction of this model is that I should be able to find states in which the domain is happy by having sparse bonds, and then there is a global minimum where all of the bonds are nicely packed together, right? And actually, if you look at the experiments carefully, so it is a system over here, this is precisely what you see. This is a domain where the bonds are fully, densely, and happily packed, And over here, in this regime over here, this is a domain over here. You see it's much lighter. This is because you're having a pattern looking much more like this.
What is
Yes.
H naught? Can you remind me what H naught is in respect to the?
This H naught is basically the position of a membrane before binding.
But why is it? So H naught is the same everywhere. At each one of those points where you have this in your top left-hand corner.
So this,
You've done a sum over all of those points.
I done a sum over all of the points, and basically this would be an H naught position. That would be an L naught position over here.
So H naught doesn't depend upon I.
The H naught is the position of the membrane in the unperturbed state. Okay, and this is basically how much, and then the deviation of that is given in this term over here. Okay, so of course after you have formed the bond, you are no longer at this position over here, but that's hidden here.
Concerning these domains, don't they have to be round?
No, they are not.
So they don't have line tension?
Okay, there we are. Good question. No. No, actually, all right, so you can calculate the line tension around the domain as a function of the size of the domain. And if the domains are fairly small, the line tension or change in the line tension by perturbing the edge is very little. And it's much smaller than the gain in the adhesion energy. Okay?
These two contributions are independent, right?
Yes, of course they are independent. And yes, you're completely right that in principle the line tension emerges, and especially emerges the larger the domain, the change is faster because the profile of the membrane changes shape as kind of if you're making around the disk, and then for very small disks basically the shape is about the same, and as you're increasing the disk it flattens and propagates deeper. But you can that can be calculated, and we did calculate that, and we know it's much smaller.
It's too small to round
It's
it.
too small to round them up. Okay, so they kind of do this. Okay, and of course that's not explicitly in the model. Okay, so and if you now run the simulations, taking into account the fact that you can't pack the bonds, then you think, okay, now you have fixed the problem, and the free energy is still extremely sensitive to the changes in fluctuations and changes in initial separation, but at least you get the phase transition line in the appropriate place.
What did you mean by simulation?
I'll talk about it in 10 minutes, but I need to introduce dynamics before. Yes?
So does one of these plots apply to the ones that are widely separated and the other one to the tightly packed ones? You had the plot before that you said...
Here this plot is done for a fixed distance between the bonds. So that's a fixed parameter for this graph. Okay.
Could you show us again the other plot?
Beg your pardon?
Could you show us again the other plot?
Yes. So this is only the effect of not looking at one, but of looking at a group. Okay, so I go from this, everything else is the same, and we've put them somewhere, so we're looking somewhere on some of these, presumably these curves, and we are probably putting them at this particular distance, right? So this is what you get. Okay? So actually the effect is quite large, the cooperativity is quite large, and you can see the changes in the plot are actually significant. They are in the order of kBt, and that whoever deals with proteins know that 1 kBt or 2 kBts are important. Okay, now a question to student biologists. What is 1 kBt? Is anybody there? I'm talking about these kBts, it's all trivial, but I mean, who knows what's actually 1 kBt?
You're asking what it represents or how big it is?
Well, no, how big it is, I'm sorry, I'm a theoretical physicist. I mean, I don't know
How big is it?
Spell out the name.
Right, it's comparable to what? Yes? is the binding energy is comparable to, if I have a binding energy of 1 kBt, this binding energy is comparable to
1 protein.
Yes, but to some, why do I have Kb and then I have T? T signifies temperature. All right, does anybody want to help?
It's relief.
Okay, so somebody from the physicist students?
I'm a mathematician.
Somebody else? So any student would like to answer this question?
Any professor.
Ooh, now
Huh? Yes?
What's the appropriate scaling for energy at a cellular level?
It's an appropriate
But
scaling of energy because this is 1 kBt tells you how much it's stored in a typical thermal energy unit. Right? So this is how much you can get out of fluctuations. Typically you get out of fluctuations 1 kBt. Right? This is at this particular temperature. So that means that if you have binding energy of 1 kBt, it means that it's comparable to thermal energy in a system. That means that thermal excitation can basically ruin it. Right? So that's why it's important to think in kBts, because it tells you, okay, if I'm solving 10 kBts, I'm an order of magnitude stronger than any thermal excitation, so I'll be stable forever. Right? Okay. So, and the reasons why we all have that, it's not only because we have shared the load, but we have also changed the fluctuations in the system. And if you look at this a little bit more carefully, then what you can see is that the membrane fluctuations will weaken isolated bonds. So if I have one bond in the membrane, and this membrane is fluctuating, the fluctuation term over there will weaken that bond. But if I now look at the membrane-induced comparativity, so not looking compared to the three-dimensional case, but looking to the case of having an isolated bond, then you will see that membrane-induced cooperativity, and this is fluctuations and bending, will strengthen the bonds. And that is why you always see domains. Very seldom, actually there's only one system where we actually saw something like a gas. So membrane elasticity is the only parameter from the physics point of view which is necessary to create domains. Yes?
Why do the domains look differently in the different proteins?
We'll get to that. All right, so they will look differently because the membrane may be different, the tension, the separations may be different, the stiffness of the membrane can be different, the nucleation time can be different, so sometimes I have one or many, depending on the nucleation time. We'll calculate all of that.
But I think this question, I'm not sure. Do you have a smaller domain, you have a bigger domain? Are you using the same cell, I hope?
It's always the vesicle. I'm not using the cell at all. This is completely passive.
Yeah,
there are no cells.
Yes, but it's the membrane that's attached to the substrate, but the domains are different.
I mean, this is one vesicle, this is another vesicle, this is another vesicle, and this is another vesicle. And I will tell you about the actual composition of the membrane. You can help me a little bit. So cadarins, and so this is two Suzanne systems. So they basically have the same membrane. This membrane is about 20 kBt in kappa. This is somewhat stiffer membrane because of larger content of cholesterol. This is Barbara's membrane. So this is 100 kBt. And in the RGD integrin system, this is Steffi's system.
Steffi or Seno?
No, this is Steffi. This is Steffi's system. Steffi's system is back basically to do the same membrane, but the lipid is somewhat different. Yes?
And then you have different concentration of the proteins?
Absolutely. That will completely change the...
Within one system?
Yes.
Yes.
If you change the
Yes,
concentration?
that will completely change the look of the domain. Okay? We'll get to that, hopefully. Now we are back. We have to think about the third process. This is diffusion. And what's the role of the diffusion in a system? The first thing which comes with diffusion is, on the thermodynamic point of view, it's entropy. All right? So I can either have a system where these receptors are immobile, or the receptors where these guys move. And if they move, or if they stop, the price of the entropy that you have to pay to form a bond is different. Here, this one is not moving, so I don't have to pay the price for arresting this one. It's already done in the preparation. I only, when I have a bond, I have to pay the price for arresting this one. If I have them mobile, then I have to pay the price for both. All right? So that is something which comes already in a thermodynamic point of view, and this is fairly powerful. And this is now what you see when you change the concentration of these guys in the membrane. Then you see that the size of the domain in equilibrium is significantly different. All right? Because the entropy-enthalpy balance overall is different. Okay, so this is systematically changing the concentration of the ligand by keeping everything else. Oh, no, basically this was, I'm sorry, changing the concentration of receptor, keeping the ligand constant. But it's the same story, because then you play with the enthalpy. Okay, if you make them mobile or immobile, then in mobile case, basically proteins can migrate into the contact zone between the adherent membrane to gain enthalpy. So the concentration will increase because we are dealing, because they just like to come in. It's good. All right? So this is then in the same system, starting from the same parameter in the immobile case and mobile case. This is kind of a filling fraction, so this is how much of this you should see in the black. You should think about it in this way. All right, so this is now two vesicles, exactly the same problem. The only difference is that here they are made immobile, and here they are mobile. That's the only thing which changes. And you see here these black points. There's very few black points. This is where the membrane is bound. This is where you have bonds. And in the mobile case, it's all of that. That's all. And it is a result of entropy.
The binding is reversible.
Yes, roughly irreversible.
Irreversible.
Roughly irreversible.
Then you would expect at the end to get the whole zone
Yes,
filled by the
yes, this is now a good question. If it's irreversible, I mean, it should be. So okay, so when we made this, we thought it's irreversible because it's integrin to RGD. So it was 10 kBTs. And if you think about the entropy, which comes from suppression of fluctuations, then it comes down. So probably is reversible. I mean, there is an exchange. You should see the exchange, and there is very, very little. It's very hard to quantify it. You should see it from the shape of the domain. All right? You should see it from the shape. So, but that's a tough question, right? Experimentally tough. Because actually to measure this, it's not so easy, right? No, I mean, how can you, I mean, you know that. I mean, what's the noise on the edge? And the noise of the edge, I mean, if you in a data analysis, is basically of the order of magnitude what you expect in the exchange. So, yes. All right. So, and that was something which kind of suggested another mechanism, which probably is not what happens here, but could contribute or surely occurs in the cell. Because this is just plain and simple physics, but is it enough or not? I have no ideas. But it basically tells you if you pull and you try to decrease this contact zone and you have mobile ligands and receptors, then your domain should increase in size. That just comes from thermodynamics of the problem. And there is a famous example where you pull on a cell and you see this adhesion point getting bigger. I guess that this is not the whole story, but I believe that this is unavoidable. This is just what happens in physics. Now, biology has to either regulate it, use it, strengthen it, or override it. But it is there, right? So you should tell me how much I'm wrong. All right.
What was the horizontal axis on the plot?
Over here, this is the size of the contact zone. How large is this area? And this is how much bonds basically you have. How much bonds do you have in a system? All right. Now we have the statics covered. We have only spoke about statics, and of course I have used more than half of my time. So I'll drive you through the dynamics. And for the dynamics, we need to introduce more. It's not enough. Let's look at diffusion first. This is the easiest bit to do. What happens when you have not one protein and then one far away, when you have many? And basically, the effect of crowding is fairly easy to understand. If you try to move through a very busy street, it's not up to you how fast are you going to move. It's basically permanently bumping into people around, and they slow you down. Right? You could, in principle, move faster, except that you can't because of all other neighbors, right? And this is now the problem of all of these people here moving together. Okay? And now, if you look at the diffusion constant as a function of concentration, this is a long time limit, then you will see that, in principle, in such a crowded environment, simple diffusion wins at long times, and you can calculate the constant, the diffusion constant which comes with that. And that was done in the 85 in a beautiful work, which somehow is not very well known for some reason.
What is CW there?
So this is the concentration of walkers.
And does it matter whether I'm a single walker moving amongst very dense barriers versus having a dense collection of walkers?
Yes, in principle, it depends. That we will talk about in the next slide. This is for dense collection of walkers. All right, so and for all of the details here, you should talk to Mislav. Mislav actually does this calculation. So what happens if I have a collection of walkers at arbitrary concentration, which walk in the arbitrary concentration of traps where they bump, and then they interact for some time? All right? So what is it that you expect? This is a problem which is old, and for some reason the long-term diffusion coefficient was not really calculated until recently. So what happens is that in a short time, as a walker, you will explore the space between the traps, and you will walk around with the effective diffusion coefficient. Depending on the concentration, this is going to be the diffusion concentration. Then you will start to explore the traps. And because you're exploring the traps, the diffusion constant, this law over here, this alpha, becomes smaller than 1. And you enter something which is called a sub-diffusive regime. And at some point, after you have explored all of your traps in your system, you swim out of this regime and you become again a diffusing particle. But you are now diffusing with some kind of an effective diffusion constant. And what I'll try to convince you is that that diffusion constant depends on the rates of interaction with the trap and your intrinsic diffusion constant. It depends, of course, also on the concentration of walkers. And this is now not a fit. This is the explicit simulation, where I think we change the concentration of walkers. And this is this formula plotted on top of the data without any fitting. So that means that now, if you can measure this diffusion coefficient over here in a long-term limit, you are not measuring your standard diffusion coefficient. You have inside embedded the fact that you're in a crowded environment and interacting with all kinds of stuff. Yes?
This can be shown rigorously, provided the time of interaction with the traps is short compared with the overall time of moving through the system.
At low concentrations, yes. At high concentrations, I did not see the calculations before. This is not fully rigorous. I mean, this is in a sense by itself a scaling argument, but a calculation. It depends here on how many guys are in the trap. So and this is done now for arbitrary number of people in the traps, which means small interactions or very large interactions, meaning I spend a very little time in a trap and I spend an infinite amount of time in a trap. And actually, the whole heart of this calculation is in calculating this bit, and that is not a trivial calculation, but we can discuss later. Okay, if you want to model binding and unbinding, then you need to put up rates into the problem. And when you are thinking about rates, then you have to come up with a model for the rates, because I mean there is a not God-given way to do the rates.
20 minutes.
Thank you. All right. There is a place to start when you talk about rates, and the place to start is from a detailed balance. Detailed balance tells you that the flux of going from a bound state to the unbound state times the probability to be in the state is the same as for the reverse process in equilibrium. And that you have no freedom, because it depends on the energetics of the system. So given that I fix the height of a member in a given spot, this is basically the elastic energy necessary to deform the receptor plus the binding enthalpy. Okay? So here you have no freedom, but now you have to come up with to break the detailed balance, which somehow is the ratio of your off rate and on rates. You have to come up with a model for that. And there is in statistical physics a standard way to do it, and we call it Cramer's rates. Cramer's rates require that you have continuous potentials, and we don't. Okay, this is the problem of having discrete potential. So then we have to fiddle a little bit. It's not exactly clean, but it still keeps the number of parameters really low. So what you have here in the exponent is the height of the barrier to go from the unbound state to the bound state, and then this prefactor has to come somewhere, and we kind of put it over here, and that is a prefactor which is hidden up here. And once you have fixed the association rate, then the off rate comes straight out, because you have no freedom, and that's it. Okay, the membrane is a healthy Hamiltonian plus the bonds. All right, and you have now propagated this Hamiltonian in time, and you can write a Langevin equation for this Hamiltonian. Oh, sorry, I forgot to. Now there is L, there is network, right? Uderam is back. So you can write the healthy, and we write it in the Fourier space. Why? Because we can do it per mode. We are dealing with the quadratic Hamiltonian. In the quadratic Hamiltonian, once you go to Fourier space, all of the modes decoupled. And actually, the propagation of the system in the decoupled modes is much faster than in the real space. So that's what we do here. So this is basically some friction coefficient, which then is in front of the this is in front of the energetics, the Hamiltonian, the change of the Hamiltonian, the derivative of the Hamiltonian, and this is basically the stochastic noise. The stochastic noise is set by the fluctuation dissipation theorem. So that was fast for anybody, but if you have questions. So, in principle, what happens? If you have strong binding, then I'm showing only the bonds. I'm not showing anybody who diffuses or not, because it would be too messy, and I'm showing only the membrane, not the underneath surface. If you have a strong bonds, then you have a domain which immediately forms, and it propagates in all happy and fine. If you have weak binding, then the situation is somewhat different. Huh? No?
It's very different.
It's very different, but in principle what you would see, bonds forming, breaking, they don't like to be stable, and then you have a large fluctuations in the number of bonds. All right. And in principle this is very nice, because now you have the full system which you can study in great details. The only problem is that you have possible length scales of 1 microns. This is how much you can treat with this system, and kind of time scales of 0.1 seconds. And I would tell you that the experiment which we looked at in the beginning took about an hour and a half. So, bummer. Nevertheless, it's still some things which you can learn. You can learn when you have weak binding. There are lots of unbinding events, and you see that things are ragged. The growth curves are ragged. They are kind of step things. This is cooperative binding and so on. Then you have strong binding that just kind of whooshes up, and then you basically kind of doop. And this is all in the reaction-limited regime, because in systems of this size, in principle, you can't speak about diffusion-limited processes. It's just nonsensical. All right? Okay. So I'll skip that. So Of course, we would like to simulate bigger systems for a long time, right? We would like to simulate the whole vesicle. And actually, it's not only my idea to simulate the big system, right? People have been trying to do this, what, for 20 years? But, what?
Forever.
Since ever. And in principle, the problem with a Hamiltonian is that you can't integrate both. You can't formally coarse-grain this Hamiltonian. You can either integrate the membrane or you can integrate the bonds. You can't integrate both. And even if you would, doesn't tell you that you would preserve the dynamics. So this is our suggestion to the problem of the solution. So rather than coarse-graining the Hamiltonian, let's coarse-grain the rates. So these are my off and on rates, which I kind of modeled or calculated on the previous slide. And I will average them by the height probability distribution at the position of the binding site. It's a very local property. Okay? At the position of the binding site, I know how to calculate my shape. I know how to calculate how much I fluctuate. That means I can know how to calculate my height probability distribution. Hence, I know how to average this. Okay? So why is this meaningful? It's meaningful because membrane fluctuations are much faster than the actual binding. So instead of calculating my rate every time for every height of a membrane, what you do is you integrate all of these motions, all of these attempts, you give them a proper weight, and you calculate the overall rate. Yes?
But does that probability distribution account for neighbors, or is that
Yes.
just a single site?
No, it has to account for neighbors. Okay, and that's precisely what's shown over here. Imagine you have this cluster of bonds. Right? This is now a cluster of bonds on a discrete lattice. In each of these squares is a bond. All right? And what I'm calculating here is this off rate. Okay? So, of course, the shape of the membrane around this guy is completely different than the shape of the membrane, and the fluctuations are different than around this guy. So the unbinding rate here is much higher than the binding here. And I have to calculate the high distribution function at this point, and at this point, and at this point, and at this point. But it seems that it is enough to look into fairly narrow environments. Right? So since it's enough to look into the fairly narrow environment, I can calculate all possible distributions and put them into a table in the end of the day. Right? And then, of course, if you would update, and you now go and say, okay, this has a probability to unbind, this is a stochastic process, some of them will unbind, even if the probability is low. This is at some later time, and you see that the probability will then change. For example, over here, I had bonds which somehow held together. I have broken these two bonds, and now this bond is also exposed, and its unbinding rate again changes. Right? And this is a way to propagate the system which now knows about the membrane, but I don't have to simulate the membrane. Okay? Now, I think I'll skip the point that these rates can be measured from kind of an average process. You have to trust me that we know that. And come immediately to that. Now, I can use this model of rates to calculate how much time does it take to make a domain? How much time it takes me to form a stable seed of the domain? So what's the nucleation time? However, before I calculate this time, I need to calculate how many bonds I need to make a stable seed. Right? And for that, we use something which we typically call a capillary approximation. And that basically tells you that you have to balance the change in the gain of the free energy with the change in the line tension. That's what it is. Right? Okay, and then from that, there is one interesting result here. It tells you that the typical number of bonds for biologically relevant parameters is very low. You need 1, 2, 3 bonds to make a stable seed. It's very low. And very quickly, if you increase these relevant parameters, then the whole thing diverges and you won't have stable adhesion. So once you have this number of bonds, you can calculate the time that takes to make these NC bonds, 1, 2, 3, or 4. And that you can do in the standard way. The way you do it in the stochastic thermodynamics is by calculating first mean passage time to have 3 bonds. This is somehow nothing new, but the point is that now you can use these rates, which are system dependent, and you know, okay, for one bond, this rate to form one bond is different because I have to go from high up and down, but once I form the bond, I have a cooperative effect, and then I can speed up the process. And then you can calculate this here. You can write this equation for something which is having, in principle, 4 bonds in a characteristic seed. And in principle, you can even solve these things quasi-analytically for a small number of bonds. And you can calculate this. And basically, you calculate this probability distribution of waiting times to form this bond, and you get something which looks like this. Now, the red curve is the actual calculation, and the black curve is coming from the longitudinal simulation where we had the rates, the small rates, explicitly. We did not integrate the rates at all. All right? So, the dots are the simulations where we did no averaging at all. This is explicit system. And the red curve is after averaging and doing the numerical calculation. Right? Okay? And it tells you, okay, that the result tells you that in principle there is a characteristic distance or characteristic sets of parameters where the bonds like to be from one another. And that somehow also agrees with the fact that there is this possibility to have dilute domains. All right? And now that you know that, you can extend the whole problem into an effective dynamics. And now you can really simulate the whole system. So now you have, rather than simulating the membrane, you integrate it out, you put it into your binding kinetics, and now you get something which you can simulate for 100 microseconds and up to minutes. You can simulate the whole system.
We have 5 minutes altogether. If there will be questions.
Yeah. No, but I mean, I think people can also ask questions as we go, right?
If you will go for another 5 minutes, there won't be time.
Do you prefer me to go for 5 minutes, or you want me to ask questions? I can then stop and say, one can do this. I can show you some examples where it works.
Just carry on.
I'll just carry on, and then you kick me out. That's it. All right. Okay, so you can show that it works, and you compare it to the higher longitudinal simulations, it works fine. And this is one system which I'm trying to show you. This is now Cadre adhesion. Now, Rudy and Suzanne and Kea have created this data 6 years ago and had it in a drawer because it looked absolutely awful. And the problem was like this. You would make one single preparation of vesicles, put them on a substrate, and observe the adhesion process. And within one preparation, you would see a process looking like this. A process where you have one domain forming and expanding. Then a process where you kind of have a lattice gas of bonds coming together. The next vesicle would be kind of few domains forming and propagating. And then sometimes even if you coarse grain, then you could have something like this. And that setting, we talked about it and had absolutely no idea. Suzanne basically given up and changed to another protein ligand receptor pair because this seemed to be way too difficult and way too irreproducible. But then we've learned what we've learned, how sensitive these bonds are to small changes in fluctuations, and how much the nucleation time can change because of these fluctuations. And we have now performed a set of simulations and calculations, calculating everything that we could. This is basically, over here is the nucleation time as a function of the fluctuation amplitude and height difference. Here we have the binding affinity in the background. Here, how many bonds do you need to form a seed? What type of the growth law you would see? You would see either a gas-like, like this, or you would see many domains like here, or radial growth like here. Depending, background is the binding affinity of the cadherin because of the environment, averaged in the equilibrium. And this is the actual simulation. And then after we did this, we came back to the experiment and reanalyzed the experiment. And what you learn is that within this small preparation, single preparation, you will just have very small variation in height. Some vesicles are a little bit larger, some vesicles are a bit smaller. The larger ones or smaller ones are a little bit higher, about 2 nanometers higher. It's enough. Or some fluctuates for 1 to 2 nanometers more or less. And that is enough to do these types of changes. Okay? And this is then for the equivalent sets of parameters. For the equivalent sets of parameters, and I put the numbers, but the B's are these ones are the D's and these ones are the C's. I'm sorry, A and B. All right. So, and then you can see that it is extremely sensitive. So, number of domains, type of the domain, the circular shape, non-circular shape completely depends on the process that you look at. You can, in the end of the day, have even things like this. This is with another system where the binding, and this is the reason for that sensitivity is that cadherins are very, very weak. They have very weak mutual interactions. So it's very quickly easy to put them in this thermal regime, thermalize them. Now, if you don't thermalize them, this is a much stronger pair. You can have something which is much larger, and I'm going to show you how the simulation grows, something like this, which is observed in the experiment. This is a jammed adhesion domain. For the equivalent set of parameters, you kind of get into this kind of a jammed state, which basically prevents. And the reason for that is that the receptors, which are trying to enter a contact zone from outside, are extremely bulky. So they're trying to enter the contact zone, they're strongly interacting with the ligand, they get clamped immediately, they create a trap. Because the trap is created, the other ones cannot move in, and basically you get stuck and you form something like this. All right? Then you can look at this in detail, build the whole phase space, and see that this kind of works. And in principle, we have picked up the right physics here. And with this, I'll conclude and say that the dynamics of the membrane with proteins can be properly constrained. We can now simulate on relevant timescales and timescales and length scales without losing the precision. That the system is incredibly sensitive to the membrane properties, and I really wonder. I really have difficulties to believe that the biology, that the cell is not using that. It's such a powerful and cheap control mechanism. You just increase locally the fluctuations. It doesn't matter where these fluctuations come from. It's a thermal, it's another process or something. You just locally increase a little bit, you make the whole thing unstable. And with this, I'll thank you. And I'll also thank my group who did all of this work. And, you can ask me many questions during the coffee break.
No question. All questions outside.
All of these.
I suggest people can leave. People who are interested in asking questions stay and ask questions.
All of these in your model. All of these binding sites are on a periodic lattice. Is that right?
So both membranes are discretized. This is not a necessity. It is making things much simpler. Right? You could do it in a non-discretized matrix and then make the diffusion really in all directions possible. It would be more complicated.
I'm thinking about your fourth order partial differential equation which
yes.
you're solving.
Yes.
Which you wrote down using Fourier. You did a Fourier analysis of the way that.
Yes. Of course.
My question is when you do compare with the experiments, are these binding sites on a periodic lattice? And the next question that I have,
No. So the receptors are not put on a periodic. So the underlying structure is a periodic lattice. So they move on a periodic lattice, but they don't. But they're not stuck on a periodic lattice. They can choose where they want to be.
Your RJs.
Yes.
But now your RJs move.
Yes. Both of them, ligands and the receptors move.
But do they move in your mathematical problem?
Well, it doesn't really matter because it's, in which one? Because there are many. So where do you want to ask? If you're
Oh,
looking.
you wrote down an equation. You wrote down an equation involving.
Okay, that's an
yeah.
equation for the membrane.
Yes.
All right. So the membrane, the way you
Which is what you're solving if you're going
Yes.
to model for the height of the membrane.
Yeah, this is what you, but you do it in 2 steps, right? You first solve for the shape of the membrane. Once you know the shape of the membrane, you look at the local rates. You choose the local rates, you try to bind or unbind every ligand receptor that is in your system. You upgrade your position of your bonds, and then once you have upgraded your positions of the bonds, you recalculate the shape of the membrane.
So you allow the RJs to move
Yes.
as they like.
Yes. Yes. Because then you propagate this in time,
So
right?
then do you have an equation for the movement of the RJs?
RJs are receptors and ligands. Yes, they move following simple diffusion.
And that's coupled to the evolution of the
Yes.
membrane.
Yes.
What are your boundary conditions for this PD?
So basically, what we solve it for the membrane in the periodic boundary conditions. When we do the Langevin simulation scheme, it's in the periodic boundary conditions, right? So everything which comes in, it comes out, right? And likewise for the membrane profile is the periodic boundary condition. Perfect. Yes?
I sort of missed, you did the master equation, and then you
Uh-huh.
have this graph of the probability of,
That's
I
here.
think, of first bond forming. Why did it have a time at which it's maximum? Why isn't it just monotone increasing?
Why is not monotonically increasing? All right, it is fairly simple. All right, this is my state P0. All right, I have 0 bonds. To move from this state, I have to, and here is my receptor, here is my ligand, right? So, my on rate has to account for this particular distance between the ligand and receptor. All right? So, see, this is basically something so I have to calculate the high probability distribution for this thing, and then integrate my K-on rate with this particular high distribution. Now, if I create my first bond, then I have done something like this. Now, this is where my next receptor is. For this particular calculation, these ones are not moving, so they are stuck. Okay, so now I have a ligand, which is, let's put this a little bit closer. All right, over here. Now I have a ligand. Now this ligand is here, but now it's much closer because of the presence of the first bond. And now I have to calculate the high distribution for this state. So here, my delta H is smaller, it's local, and my fluctuation amplitude is also smaller. All right, because here I'm suppressing fluctuation.
What does that P represent precisely?
So I'll get to that then. So basically that, now so here I am unbinding with this off rate 1, having 1 bond. And here now I'm binding by having 1 bond, and I'm creating another K on 1. All right? And now I have to put the next bond, put it over here. But then now I have a profile looking like this. Okay? So now what I see is that this profile is basically very similar to this profile, and that every next bond will be created with the same on rate.
Well, I thought that was the probability of the first bond forming.
This is the probability to form the first bond, which is clearly different than the probability to form the second bond. Right?
But again, what does the P of T represent?
This one?
Yeah.
This is basically the first mean passage time. So how much time do I need?
Oh, for n bonds.
Yeah.
Okay.
Okay.
I thought it was for the first one.
No, this is the only thing I'm trying to
For
say
the
is
first
that
n.
yes, no, for the P node, but the P node, the rates for the P node state are completely different than the rates for any other state. And that creates you another monotonic distribution function.
I suggest that now we've got the discussion.
We can go on to the next talk. Take your time and chairman.